Genes that encode various proteins determine the structure of an organism. Therefore, alterations in the genome of an organism may result in new traits. For example, it is possible to produce sheep with a fluffier fleece. The first organisms into which foreign genes were incorporated were bacteria. New genes are incorporated into bacteria by means of plasmids, small circular DNA molecules, each of which has its own origin of replication. The gene that is to be introduced into the cell is incorporated into a plasmid. Plasmids modified in this way are then incorporated into bacterial cells. The bacteria absorb the plasmids directly from the medium. This is how transgenic bacteria are obtained. In order to distinguish the plasmid-containing bacteria from those without plasmids, marker genes, which are usually genes for antibiotic resistance, are introduced into the plasmids. Bacteria that contain plasmids with an antibiotic resistance gene can survive in a medium containing the antibiotic. On the other hand, bacteria that do not have this plasmid die. Transgenic bacteria are used in the synthesis of biologically active proteins, such as insulin or growth hormone. They are also used as manufacturers of great numbers of copies of the incorporated gene or plasmid. These copies are then used to study gene properties.
Thank you. Today, we start a new topic, transgene and future prospects of transgene under the chapter 3, plant genetic transformation prospects and potential. So first we see what is transgene. Transgene is a gene that has been transferred naturally or by any of a number of genetic engineering techniques from an organism to another. When we will transfer any specific or any desired genetic material or a specific gene from one species to another species by naturally or any genetic engineering technique. It may be direct or indirect methods such as biolistic, agrobacterium mediated transformation. This type of gene, this type of specific gene or this type of desired characteristics is called transgene. Future potential application of transgene. The study of application of transgene is a rapidly growing area of molecular biology. In fact, it is predicted that in the next 2 decades, 3 lakh lines of transgenic mice will be generated. Researchers have identified many applications for transgene, particularly in the medical field. Scientists are focusing on the use of transgene to study the function of human genome in order to better understand disease, adaptive animal organs for transplantation into humans, and the production of pharmaceutical products such as insulin, growth hormone, blood, anticoagulating factor from the milk of transgenic cow. So, with the research of transgene, we will develop transgenic mice for the study of different types of production of different types of pharmaceutical product, insulin, growth hormone, blood, anticoagulating factor, interferon, and different types of biotechnology product globally, different types of vaccine also product and evaluation by using this type of transgenic mouse, and also we also use this type of transgenic mice for the treatment or for the evaluation of the status by using this type of transgenic mouse, such as diabetics and also different type of genetic abnormalities. Also, there are currently 5,000 known genetic disease, and the potential of treat this disease using transgenic animals is perhaps one of the most promising application of transgene. There is a potential to use of gene therapy to replace a mutated gene with a unmutated copy of transgene in order to treat the genetic disorder. So, by the use of transgenic mice, we also treat a genetic disorder by the replace of a normal gene. This therapy is called gene therapy. This can be done through the use of Trelox or knockout approach of genetic engineering. Moreover, genetic disorders are being studied through the use of transgenic mice, pigs, rabbits, and rats. More recently, scientists have also begun using transgenic goats to study genetic disorder related to fertility. Transgenes may soon be used for genoplantation from pig organs. Though the study of organ rejection, it was found that an acute rejection of the transplanted organ occur upon the organ's contact with blood from the recipient due to the recognition of foreign antibodies or endothelial cells of the transplanted organ. Scientists have identified the antigen in pig that cause this rejection and therefore are able to transplant the organ without immediate rejection by removal of antigen. However, the antigen being expressed later on, the rejection occurs. Therefore, further research is being conducted. So, by the use of transgene, we also conducted the new era transplantation. We know presently different types of organ, heart, kidney, liver, and also different animals organ now able to transplanted it on the basis of the antigen or antibody activities. This examination also evaluated by the use of transgenic mice and also the transgene. Transgene are being used of manufacturers to produce goods such as milk with high level of proteins, silk from the milk of goats, and microorganisms that are capable of producing proteins that contain enzymes that increase the rate of industrial reaction applications. Industrial applications aim to selectively breed animals for particular traits and animals that are resistant to disease. So, by the use of transgene, we also produce different types of transgenic animals, such as goat, cow, sheep, and others transgenic animal that will provide a high level of protein from milk or meat. So, you read future potential applications of transgene in this area, only 5 to 6 points. No need read detail, you just mark only 5 to 6 points. 5 to 6 points